PLB112. Our topic today that we'll be presenting is sudden oak death. As the name suggests, it's a disease that negatively affects oak populations in California and Oregon forests. Ultimately, it can kill these trees. The pathogen behind this is called Phytophthora ramorum. It is a water mold pathogen that causes symptoms such as leaf spots, twig dieback, and bark cankers. The origin of its name is pretty straightforward. When it was initially discovered, it was named for its lethality. The rapid deterioration of the tree's health without prior illness, accompanied with the browning of the leaves, worried many. They noticed the difference between the supposed sick leaves and regular healthy leaves with sunburn damage. To the left, we can see a variety of oak leaves that are completely healthy, and to the right are images of how browning on oak leaves would look like if it was caused by P. ramorum.
P. ramorum grows on leaves of host oak species, and its spores travel through water. We will go in more depth about this soon. In addition to oak species, P. ramorum affects more than 75 different plant species. This is a very serious issue. P. ramorum is considered a generalist pathogen, which means it is a pathogen that has a wide range of hosts. In fact, it has over 100 known host species. Its main host is tan oak. The reason why this pathogen is able to have so many different hosts is due to its diverse phenotypic variation, which results in varying degrees of colony morphology, senescence, and virulence. Studies have shown that this is in large part due to the fact that many plant pathogenic genomes, such as P. ramorum, are composed of transposable elements. Transposable elements are sequences of DNA that are capable of changing their position within a genome. Researchers therefore believe that their observations are in favor of the epitransposon hypothesis, which is the hypothesis that epigenetic regulatory mechanisms such as RNA interference, DNA methylation, and histone modification suppresses transposable elements. However, when P. ramorum are actively colonizing oak stems, physiological stress is triggered, which disrupts epigenetic suppression of transposable elements. This allows the transposable elements to reactivate and lead to possible genome diversification. On the right, this is an image of some of the characteristics of transposable elements.
Here is a list of just a few out of the many plant species that are negatively affected by P. ramorum according to the U.S. Department of Agriculture. You can pause the screen here if you're interested in the other pathways, but for our presentation, we'll focus on oak species. P. ramorum invades healthy trees via the natural openings in the bark. As it accumulates and grows, it begins to kill the cells of the localized area. This can inhibit water and nutrient transport, slowly making oak trees more and more sick.
Now, let's get into the life cycle of the pathogen. This is an image from Washington State University that gives an overview of it. We can see that on the right of the image, there are many pathways that P. ramorum goes under to reproduce. However, we will focus on just one of them. There are two cycles in which P. ramorum disperses itself. First, there's asexual reproduction. The surface of infected leaves and twigs hosts sporangiophores. A structure called the sporangium produces zoophores that can invade the host and start to colonize within. There's also a hypothesized cycle of sexual reproduction that takes inspiration and inferences from the disease cycle of P. infestans. On the infected leaf, a separate structure called the oogonium undergoes meiosis to produce oospores. Now we will dive deeper by going more in depth about a proposed disease cycle for P. ramorum. This is how P. ramorum will affect tan oak. An external source of P. ramorum can infect a host, in this case, California bay laurel. This host in close proximity can infect tan oak trees that begin to develop bark cankers. As the tan oak tree grows, its leaves may fall to understory trees and shrubs, and even to piles of leaves on the soil. The pathogen can then sporulate in the leaves and further infect other tan oak trees.
How then does P. ramorum get transmitted? The natural dispersal is through the transport of infected plant material, which is mainly done by wind-blown rain. Another potential pathway is through the dispersal of waterborne and soil-borne chlamydospores, which are thick-walled spores that are able to survive extreme and harsh conditions. It turns out that humans can also be a potential vector through the displacement of contaminated soil, water, and leaves by means such as clothing, vehicles, and equipment. Other potential vectors are birds, mammals, and insects. However, more research needs to be done to examine this. Infection by P. ramorum can lead to 3 distinct types of disease syndromes. They may share similar characteristics. The first one is our topic, which is sudden oak death, which is mainly characterized by cankers. Cankers are sores or wounds on barks and branches. They are usually discolored and may often be bleeding. This bleeding is due to dark red to black sap oozing from the trunk. Sometimes cankers may also have a fermentation smell, which smells like alcohol. Another disease syndrome that can be characterized by this pathogen is Ramorum shoot dieback, which is mainly characterized by leaf blight and branch dieback. Leaf blight can be caused by water-soaked lesions, while branch dieback is progressive death of branches or twigs that start at the tips. This can lead to girdled trees and wilted leaves. Another disease syndrome is Ramorum leaf blight, which is mainly characterized by leaf spots, blotches, and sorches. These usually result from water accumulation on the leaves. However, the twigs and branches are usually unaffected. In this image, A is an example of a bleeding canker of tan oak, B are leaf spots, and C and D are leaf necrosis.
Early in the discovery of the disease, potentially infected trees were tested using molecular diagnostics. Otherwise, they could already observe symptoms that were presenting itself on leaves, twigs, and bark. Now, there may be another approach. Catherine J. Hayden and her colleagues developed a method using polymerase chain reaction, PCR. They used primer pairs that were specific to P. ramorum, Phyto 1 and 4, and Phyto 2 and 3. The data from their study showed that PCR was more sensitive than other tests, as it could detect a lower level of infection. However, it is limited by the window of optimal testing from March to June. Also, this method has a possibility of producing false positives. Despite this, they're enthusiastic about the results of the study, as it could potentially lead to the discovery of new hosts and symptoms of P. ramorum to better detect and address this disease.
Unfortunately, there is no cure for sudden oak death. Preventative measures, however, can be taken. For instance, before nursery plants are introduced to an area, they should be inspected for the disease as they can host the pathogen. If an infected tree is found, they must be removed from the area along with any debris that are left behind. The area must also be thoroughly sanitized, as any bit of soil or leaves can migrate to an uninfected area. Lastly, there's a phosphonate fungicide named Agrifos that can be administered to highly valued oak and tan oak trees. This treatment must be done before infection on healthy trees and maintained every so often, such as 1 to 2 years with a booster. It can also act to alleviate some symptoms that infected trees experience. So why is all this so important? Well, there's a lot of diversity in forests. We need it to filter water, to cycle nutrients through the forest, and to form soil. When there is chemical and nutrient imbalance, we risk altering decomposition rates of trees. The frequency and intensity of forest fires would be increased as well. Forests are also sources for shelter and food for animals. It is estimated that sudden oak death has killed over 1 million trees in California alone. Just look at this slide. Look at the many variety of California forests. There's redwood forest, pine forest, conifer forest, and oak woodlands, which are all at risk of undergoing detrimental decline if the pathogen is not controlled. Luckily, we have amazing researchers, such as the ones at the University of California, that are currently working hard to discover and develop new ways to tackle this issue. Thank you for listening to this presentation, and the next time you walk by an oak tree, you just might want to examine it